Hello guys, I am Kavita and I welcome you all to my YouTube channel Exploring Pharma. If you are new to my channel, please subscribe and press the bell icon to get notifications of all my new videos. In today's video, we are going to explore about coronavirus, that is COVID-19. What is COVID-19? Coronavirus disease is a disease which is caused due to newly discovered coronavirus. Coronavirus is an contagious disease which affects mammals, that is human beings, animals as well as birds. Coronavirus disease is caused due to severe acute respiratory syndrome, that is SARS-CoV-2. Coronavirus was first reported in bats and the first case was identified in Wuhan in China in December 2019, and since that time the coronavirus has entered or spread all over the world, leading to a pandemic. 15.7 crores cases reported till now, of which 9.3 crore people have recovered, whereas 32.8 lakh people have died. Symptoms of coronavirus are variable, but most common symptoms include fever, chest pain, difficulty in breathing, fatigue, and loss of smell and taste. Symptoms may develop 1 to 14 days after exposure to coronavirus. A third of people who are infected with coronavirus do not show any noticeable symptoms. Now we will discuss about life cycle of coronavirus. Now let's see the structure of SARS-CoV-2, that is coronavirus. Coronavirus is derived from Latin word corona, meaning crown or wreath. What is wreath? It is something arranged in circular shape as a decorative arrangement of flowers in it. As you can see, the coronavirus structure consists of arrangement like flowers on it. Now, what the structure of coronavirus consists of? It consists of lipid bilayer. Lipid bilayer are the two layers which are made up of lipid, which forms continuous barrier around virus. You can see this black lining which forms a layer around virus. Now, coronavirus consists of RNA genome, that is single-stranded positive sense RNA, that is plus ssRNA. Coronavirus consists of 4 structural proteins, one of which is nucleocapsid protein. What is the function of this nucleocapsid protein? It is involved in packaging of RNA genome and it is always present with the genomic RNA. You can see this pink part is the nucleocapsid protein and this blue part, which is in coil form, is genomic RNA, and this nucleocapsid protein is present with the genomic RNA, forming a kind of complex. The next protein is spike protein, denoted by S. Spike protein functions in facilitating virus attachment to ACE2 receptors present on host cell and helps in fusing of virus with the membrane of host cell or the human cell. The third protein is the membrane protein, denoted by M, which is responsible for shaping the virion. And the last protein is the envelope protein, denoted by E, which is responsible for virion assembly and its release. Now we will see genome of SARS-CoV-2 or coronavirus. Coronavirus contains single-stranded positive sense RNA genome, that is plus ssRNA. What is RNA? RNA is ribonucleic acid and it is made up of nucleotides such as adenine, guanine, cytosine and uracil. RNA has only one strand. What is genome? Genome is organism's complete set of genetic instructions, and each genome contains all information needed to build that virus and allow it to grow and develop. Coronavirus has largest known RNA virus with the size ranging from 27 to 32 kbp, that is kilobase pair. Kilobase pair is a unit used to measure the length of nucleic acids because genome is made up of nucleic acids only. Coronavirus genome encodes 5 major open reading frames. As you can see, this ORF, and here what are this ORF? ORF is open reading frame and it is part of reading frame that has ability to be translated. What is translated? It is a translation process in which proteins are created from mRNA. Okay, so this coronavirus genome encodes 5 major open reading frame ORFs including two polyprotein, that is ORF1A and ORF1B, which is present at the 5' end of this genomic structure, and it also encodes four structural proteins, that is spike protein, envelope protein, membrane protein and nucleocapsid protein, which is present at the 3' end of RNA genome of coronavirus. Now this ORF1A then is further consist of non-structural proteins, one of which is non-structural protein 3, which denotes papain-like protease, which is PL-PRO, and the other is non-structural protein 5, which denotes 3CL protease, that is 3CL-PRO. The other open reading frame 1B consists of further two non-structural proteins, which are denoted by NSP12, that is non-structural protein 12, which indicates RNA dependent RNA polymerase, which is RDRP, and the other one is non-structural protein 13, which indicates helicase. Now this all are non-structural proteins, and the rest of these four are the structural proteins. Okay, spike protein, envelope protein, membrane protein and nucleocapsid protein are the structural proteins. And structural proteins are involved in receptor recognition, virus attachment, penetration, membrane fusion and virion assembly. Now let's see life cycle of coronavirus. The first step which is involved in life cycle of coronavirus, we will see or we will discuss about that in detail now. This is cross section of spike protein. As I explained, these are the spike proteins which are present around coronavirus. Coronavirus are surrounded by that spike protein. This is cross section of that spike protein. As you can see, this is the whole spike protein. Okay. It is divided into two parts, S1 which is the upper part and S2 which is the lower part. Okay. What is the function of this S1? S1 helps in receptor binding, that is it will bind to the receptor which is present on the host cell, which is ACE2 receptor, angiotensin converting enzyme 2 receptor. S1 will bind to specific receptor which is located at the surface of host cell. Okay, and it consists this receptor binding domain. Okay, the next is the S2, which functions in mediating fusion between viral envelope and target cell membrane. Okay, this is responsible for binding and S2 is responsible for fusion. Now between this S1 and S2, a cleavage site is present as I have shown it here. Okay, now what happens is that this is host cell containing ACE2 receptor, as I said, angiotensin converting enzyme 2. Whenever a virus containing spike protein and other proteins as well, but here we are discussing mainly about spike protein. Whenever a virus containing spike protein will come, the spike part of that coronavirus will attach with this ACE2 receptor and the S1 part will bind. Okay, because S1 is functioning in binding. And what happens is that, as I said, there is cleavage site present between this S1 and S2. It will be cleaved or braked with the help of this protease enzyme called furin and TMPRSS2. These are the two enzymes which will break S1 and S2. What will happen next is that then the S1 part with the ACE2 receptor will then go out, and then this S2 part will go and bind with the host cell. Okay, and with this process the virus would be able to enter into the host cell. Now we will see the further process in life cycle of coronavirus. You can see here, this is the host body containing host cell. Whenever a person, that is if suppose this person is infected with coronavirus, whenever this person will sneeze, cough, speak or sing, droplets from his mouth will act as agent which will help coronavirus to, these droplets will help coronavirus to enter the other host cell. This is the host cell of other person, from his eyes and nose and mouth. Okay. Then this is the cell. This is the cell which is present onto the alveoli of our lungs. Alveoli is the part of our lungs where gas exchange takes place. So on the cell, ACE2 receptors are present. You can see here this is ACE2 receptor. Okay, ACE2 receptor act as receptor for SARS-CoV-2 virus and allow it to infect the cell. Now what will happen is that, as I explained earlier, spike protein of coronavirus, it is this spike protein of coronavirus, it consists of two proteins S1 and S2. So when S protein, this part, the whole part of coronavirus does not bind with the ACE2 receptor. Only the S protein part binds to the ACE2 receptor. When this binding takes place, TMPRSS2, that is transmembrane protease serine 2 precursor, and furin, they both will go and cleave or break the spike protein and they will activate the spike protein. Basically, some proteins are in the inactive form and must have sections removed to become inactive. So this TMPRSS2 and furin will help in the activation of that spike protein. The next step is that fusion and entry with the process called endocytosis. Endocytosis is the process in which virus is brought into the cell. Okay. Now the virus has entered into the cell. Now what happens is that an endosome layer will cover this virus. Okay. What this endosome will do, it will provide an environmental condition for virus and also it will provide platform for virus replication. The next step is uncoating. In the uncoating process, the RNA genome, as you can see, the coiled part will get removed from the virus and it will go in further process, which is translation. You might be wondering, what is translation process? Translation is the process in which proteins are synthesized from RNA. Okay. Next question will come to your mind is that why protein? The answer to this is that as coronavirus contains protein in its structure, the main aim of coronavirus, which has entered into the host cell, is to multiply more coronavirus. For this process, the new coronavirus will require protein, and for that the coronavirus makes protein. Okay. Ribosome, that is the host ribosome, is responsible for protein synthesis. This protein synthesis process takes place in ribosome. Ribosome is an organelle present in human cell. Okay. And SS genome RNA, which is the genome of coronavirus, then form replicase polyprotein. Okay. Which replicase polyprotein? PP1A and PP1B. Now, this replicase polyprotein will then undergo a process called proteolysis. What is proteolysis? It is breakdown of protein. The word says breakdown, lysis of protein, which will be done with the help of 3CL-Pro and PL-Pro, as I explained earlier. This is the non-structural protein part of RNA genome. Okay. So then the replicase polyprotein will then form 16 non-structural proteins from 1 to 16, of which one is RNA dependent RNA polymerase, that is RDRP. What this RDRP does is that it is used to synthesize negative strand using positive strand of coronavirus as template. Now the question will come to your mind, why negative strand of genomic RNA of coronavirus? First, let's see that what this 16 non-structural protein will do. They will form RTC, that is replication transcription complex. The word itself says replication transcription. Replication means duplicating and transcription is the process in which RNA produces messenger RNA. Now as we will go further, you will understand what is this replication and transcription process and why they happen. Okay. So this RTC complex will then undergo replication process in which the positive strand of coronavirus will get converted into genomic RNA minus. Okay. And in this minus strand, it consists the orientation in the form 3' to 5' and positive strands cyst of orientation 5 dash 3 dash. Okay, they are opposite. That is why this is positive and this is negative. Now this negative genomic RNA will then undergo a process called transcription. Okay, as I said, transcription is the process in which RNA will form mRNA. As you can see here, mRNA is there. Okay, but this process is not direct. There is one more step involved, is that this genomic RNA will form first subgenomic RNA negative. What is subgenomic? Subgenomic RNA are smaller sections of RNA. Okay. Now this subgenomic RNA negative will then form subgenomic mRNA positive, and this subgenomic mRNA is required for production or synthesis of 4 proteins which are important in the structure of coronavirus. As I explained, the 4 proteins are spike protein, envelope protein, membrane protein, and nucleocapsid protein. So how the synthesis takes place? It takes place with the help of subgenomic mRNA with the process called translation, and as translation is preparation of synthesis with the help of mRNA. So you all would have understood why all this process takes place, why this positive is converting into negative RNA, because it has to be then converted into positive subgenomic RNA, because all this protein synthesis will happen only when subgenomic mRNA is present. Now out of this whole protein, this nucleocapsid protein, what it will do, it will go and bind to the Golgi. Okay, only the nucleocapsid protein will go and bind to the Golgi, which is also an organelle of cell. Now at this site also the positive genomic RNA of coronavirus, which is the important ssRNA positive, which is required in coronavirus, this will also go and bind to the nucleocapsid protein and they will form a complex there. Now this 3 remaining protein will then go to the endoplasmic reticulum. Okay, the endoplasmic reticulum is also a cell organelle. From here, this 3 proteins, as you can see, this spike protein, this green one is envelope protein, and this is the membrane protein. All the 3 proteins will then go to the site of Golgi from endoplasmic reticulum to Golgi, and they will form endoplasmic reticulum Golgi complex. Okay. Now, as you can see here, all the parts of coronavirus are present. The genomic RNA, nucleocapsid protein, spike protein, envelope protein, membrane protein. So what will happen is that assembly, all this part will assemble themselves in all the perfect positions, that is in round shape, and through Golgi vesicle, with the process of exocytosis, the virus will go out of the host cell. Now a very active fresh coronavirus is prepared, which will then enter the other host cell. As you can see, it will enter the other host cell, and the same process will then happen in that host cell bodies also, and then that person will then transfer coronavirus to other person, and in this way this cycle or chain of coronavirus goes on and it keeps infecting people. Now we will see about pathophysiology of COVID-19. In previous slides we saw about action of coronavirus at cellular level. Here we will see that after affecting the cell of alveoli, what it does further. So this is human lung, which consists of trachea, and there are branches present, and the end part is alveoli. Okay, and which contains alveolus, and this is the place where COVID-19 or coronavirus will affect. What happens is that alveolus are responsible for exchange of gases, that is they absorb oxygen and they release carbon dioxide. As you can see here, there are 2 types of cells, that is type 1 alveolar cell and type 2 this blue color. 2 cells are present which are called alveolar cell. Okay, and the function of type 1 alveolar cell is that they are thin enough that the oxygen passes from alveolus to the blood capillary. Okay, the function of type 2 alveolar cell is that they secrete surfactant, which is a substance that lines alveolus and prevent it from collapsing. This is the line of surfactant which is preventing alveolus and blood capillary, that is, it is maintaining a barrier between both of them. This is the function of surfactant. Now what happens is that, as I explained earlier, when the spike protein which is covering coronavirus will bind to ACE2 receptor which is present on this type 2 cell, they will allow the virus to inject its RNA into the host cell. The RNA will then hijack the cell and it will tell it to assemble many more copies of virus and release them into the alveolus, and the host cell is destroyed in this process and the new coronavirus infects neighboring cell. Okay. So what happens is that in normal person this all processes in the normal concentration, but when a person gets infected, what happens is that the fluid get develops into the alveolus due to release of cytokines, macrophages, inflammatory signals, due to which there is reduction in the exchange of gases, and this alveolar type 2 cell is also infected, due to which there is reduced surfactant which will lead to collapse. That is, pores will develop into the lining of alveoli, due to which all the components between alveolus and blood capillaries will exchange. And in severe conditions, what will happen is that this protein rich fluid which has accommodated into the alveolus will then increase, and also there will be more pore formation, as you can see here, due to which all the protein rich fluid will enter into the blood and it will enter to whole part of body. Now we will see next, that is immune response of host body. As I explained, when coronavirus affects the type 2 cell, they release inflammatory signals which will recruit macrophages. That is, they will make more macrophages to come at that place, and these are immune cells. Macrophages will release cytokines which will cause vasodilation, which will allow more immune cells to come at the site of injury. If any person is in this stage, he or she will experience symptoms like dry cough and fever. At the next step, as I explained, the fluid will accumulate inside the alveolus. If any person is in this stage, he can develop pneumonia. Pneumonia is basically a disease in which person's alveolus get filled with protein-rich fluid, due to which there is diminish of oxygen and carbon dioxide exchange. The next step is that this fluid, the protein-rich fluid, will dilute the surfactant, which will cause alveolar collapse, which will decrease gas exchange and increase work of breathing. That is, there will be pore formation between the lining of alveolus and blood capillary, due to which the surfactant will get diluted and the person will require more oxygen, due to which there will be shortness of breath. Next step is that neutrophils are recruited. That is, neutrophils are also brought to the site of infection, which will release reactive oxygen species to destroy the already infected cell. If any person is in this stage, he or she needs to be hospitalized. The next is that type 1 and type 2 cells are destroyed. They are fully destroyed, leading to collapse of alveolus, causing acute respiratory distress syndrome. If any person is in this stage, he needs to be shifted to ICU and the patient may require ventilator and life support. Okay. Now the very severe step occurs due to when inflammation becomes severe, which is when the protein rich fluid enter the bloodstream, which will then distribute whole over the body and which will cause systemic inflammatory response syndrome, SIRS, and this may lead to septic shock, multi-organ failure. That is, many organs will get failed and this will eventually lead to death. And if any patient is in this stage, complications other than COVID-19 may occur, but with proper care, patient may recover at any point during this process. I hope you find this video helpful. If you like this video, don't forget to like, share, subscribe and comment. In next part, we will see about coronavirus in more detail, that is about vaccines, preventions and tests, etc. Stay tuned and updated. Thank you.